Our research is concerned with modeling protein DNA complexes, specifically those that involve the protein AraC. AraC is composed of two subunits, the dimerization domain, shown here, and the DNA binding domain, shown here, which binds to the DNA at three specific half sites, I1, I2, and O2. The structures of the DD, DBD, and DNA are known independently, but the structure of the whole AraC and DNA complex is not. Our goal was to use software to create a whole AraC DNA complex that models their natural interactions. For our research, we used known X-ray crystallography structures of the unbound apo form of AraC and the arabinose-bound holo form of AraC. We also used a known NMR structure of the dimerization binding domain. In addition to these proteins, we also generated straight DNA of the I1, I2, and O2 half sites. To begin with, we attempted to dock these straight models to the DBD. For docking the DNA half sites to the DBD, we used the HDock software program. This program tests many different orientations of the DNA-DBD binding and scores the top 100 docks, with a more negative docking score representing a more favorable dock. In addition to looking at the docking scores, we also had to ensure that the DNA and DBD were bound in the correct directions, that the DBD bound in the major grooves of the DNA, and that the known biochemical contacts were bound. Shown here is a preliminary model using HDock of straight DNA bound to DBD at the I1 DBD complex. This is the 5' end of the DNA, and this is the 3' end of the DNA. There should be a dock here, but as you can see, no contact is being made between the DNA and DBD. Unfortunately, it was difficult to achieve a viable dock with straight DNA. Moving forward, we used a variety of methods to manipulate the DNA and create a better dock.
The first method we used to create a better segment of DNA was the implementation of a genetic algorithm, which involved defining 6 overlapping sequences of DNA in each half site and finding existing structures that matched with these segments. These were found in the RCSP database, which contains many examples of known DNA structures. For each of the 6 segments, 4 examples were found and had their parameter values averaged. Finally, these averaged segments were pieced together into the I1 and I2 half sites. We hoped the use of natural DNA structures would create a more realistic half sites for both and achieve a better dock. While docks were better after implementing the genetic algorithm, further DNA bending was required for each half site to create an optimal model. For the I1, I2, and O2 DNA half sites, we implemented a method to manipulate the role parameter between each base pair outlined in Understanding DNA by Calladine. In nature, DNA strands are not rigid. They can conform to the shape of the protein they are binding to. Calladine describes the use of a cosine curve to introduce a natural bend to the DNA so that the known DNA-DBD contacts in the DNA half sites can dock successfully in HDock. With this cosine method, the DNA strands are in a much more ideal position to successfully bind to the DBD protein. The cosine role manipulations were made using Web3DNA software.
Shown here is the same I1 DBD complex shown previously, but as you can see, the large gap has mostly been removed, and an important biochemical contact between the DNA and DBD is now made that was not previously made.
After we had successfully created a model for the I1 and I2 half sites with their corresponding DBDs, we moved on to modeling the full structure of AraC. A full segment of DNA containing both modified half sites and the 4 base pairs between them was created. The DBD I1 and DBD I2 complexes previously described were aligned with this new long segment of DNA. The structures were merged to create a segment of DNA with both DBDs attached. With the help of HDock and PyMol, we were able to dock this new structure with the dimerization domain. However, one of the DBDs was positioned too far from the DD, so using the cosine role method, it was necessary to bend the full segment of DNA with DBDs attached to create a viable model.
We researched the complexes created by DNA and the DBD at the 3 binding sites I1, I2, and O2. While we did achieve docks in both of the major groove regions of our DNA molecules, it is important to note that protein shape can also change to create a better, more favorable dock. In reality, the DBD and the DNA likely conform to each other, making a stronger dock. Using these models, we were able to create a model for the AraC protein in the holo form while bound to the I1 and I2 segments of DNA. There is a concurrent experimental effort at Loyola using the atomic force microscope to measure DNA bending around AraC. Ideally, the computational models and experimental results will support each other.